Cells involved in anti-cancer response. Several immune cells respond to cancer or tumor cells, forming the immune surveillance system that detects and destroys abnormal or transformed cells before they form tumors. The key immune cells include number 1, the natural killer cells, which directly kill tumor cells without prior sensitization. These cells release perforins and granzymes to induce apoptosis in cancer cells. Therefore, the natural killer cells are part of the innate immunity and act rapidly against abnormal cells. Number 2, cytotoxic T lymphocytes, the CD8 T cells, specifically target and kill tumor cells displaying tumor antigens on MHC1 molecules. These cells are activated by antigen-presenting cells, releasing perforins and granzymes, which induce apoptosis in the target cell. So the CD8 T cells are part of adaptive immunity, providing specific and long-term response. Number 3, helper T cells, the CD4 T cells. These indirectly support anti-tumor response. They activate the CD8 cells and macrophages through cytokine release, for example, interleukin-2, and stimulate B-cell antibody production. Therefore, they coordinate and amplify the immune response against tumor cells. Number 4, macrophages, majorly the subtype 1, which kill tumor cells and present tumor antigen. They release reactive oxygen species and tumor necrosis factor alpha and also present antigens via MHC2 T cells. So the macrophages provide both innate killing and bridge to adaptive immunity. However, some macrophages, majorly the subtype 2, can promote tumor growth. Therefore, macrophages can be pro or anti-tumor depending on their activation type. Number 5, dendritic cells. Prime T cells against tumor antigens. They capture tumor antigens and migrate to lymph nodes, present antigens via MHC2 and 1 to CD8 and CD4 T cells. Therefore, they are essential for initiating tumor-specific adaptive immune response. Number 6, the B cells, which are less directly involved. The B cells produce antibodies against tumor antigens, recognize soluble tumor antigens and present them to T cells, and the antibodies may lead to antibody-dependent cellular cytotoxicity. Therefore, they may enhance immune response or modulate tumor environment.